Hello dear learners. In this video we are going to learn about the two important effects, one of them being the cytoplasmic inheritance and the another one being that the maternal genetic effect. So first of all, let us start with the topic of cytoplasmic inheritance. Now what is the meaning of cytoplasmic inheritance? This phenomenon of cytoplasmic inheritance is also referred to as the extranuclear inheritance. We know that in our cell there is a nucleus, and this nucleus contains our genome or DNA, and this DNA consists of the genes. Now when the particular organism has to reproduce, these genes in the haploid set will move into their gametes, and then similarly from another parent also the haploid set of genes will come into their gamete, and these will fuse to form a diploid zygote. Now this is a very typical process wherein the haploid side comes from the maternal side and the another haploid side comes from the paternal side, and after that there is a diploid zygote which is formed. Now this is what we call as the nuclear inheritance. So whatever are the genes which are present on the DNA of the parent and the including mother and the father, they will be inherited into the offspring. This is the nuclear inheritance, but when we talk about extranuclear inheritance, it refers to all those genes inherited which are not present inside the nucleus or which are not present in the nuclear DNA. Can you guess of any other place where the DNA is present except for nucleus? So yes, the DNA is present in two specific organelles, that is mitochondria. In mitochondria it is known as empty DNA, and then there is also chloroplast, and in chloroplast this particular is called as cpDNA. So in mitochondria it has its own DNA which is referred to as the empty DNA or the mitochondrial DNA, and similarly the chloroplast consists of the cpDNA. So when this empty DNA or this cpDNA will be inherited to the offspring, this will be referred to as the extranuclear inheritance or simply the cytoplasmic inheritance. Why we are using the term cytoplasmic? Because the mitochondrial DNA and the chloroplast DNA are located inside the cytoplasm in the organelles including mitochondria and chloroplast. Now there is one very important process here which follows. Whenever the egg or the ovum and the sperm has to fuse, always remember that from the sperm only the nuclear elements will be contributed, but from the ovum other than the nuclear elements whole of the cytoplasm is of ovum. So ovum will contribute both nucleus as well as the cytoplasm to the resultant zygote, but the sperm will only contribute the nucleus. So in that condition, always remember that the cytoplasmic components will always come from the maternal side. So when we talk about the cytoplasmic organelles and when we talk about those DNA which are present in the cytoplasm, they will be coming from the mother, or they will be simply coming from the maternal side. So whenever the cytoplasmic inheritance has to happen in the progeny, the inherited genes will always come from the maternal side except for some exceptions. So this is one of the very important concepts which you should know about the cytoplasmic inheritance. Now let's move on to the examples here where we can have the discussion on that. So yes, please remember that if I talk about humans, in humans the mitochondrial genome, that is the genome of mitochondria, consists of approximately 15,000 nucleotides only. Now this is very small as compared to the DNA which is located in the nucleus, and it only encodes for about 37 genes. Now please remember, one cell can contain many mitochondria, and one mitochondria can contain many DNA, similar DNA, that is many mtDNAs. mtDNA. So what I mean to say is one cell can contain many of these mitochondrial DNAs. So if we compare these mitochondrial DNAs with the nuclear DNA, we come to know that the nuclear DNA encodes for about 20,000 to 25,000 genes, but the mitochondrial DNA is only encoding for 37 genes. In addition to that, we also come to know that there is chloroplast which is present in the plants. These chloroplasts also contain the DNA which is known as the cpDNA. Now the cpDNA will also be present in the cytoplasm, and it will also follow the pattern of the cytoplasmic inheritance. When a zygote is formed from the maternal ovum and the paternal sperm, this zygote will inherit the nuclear genes from both the parents. But all of the cytoplasmic organelles of or in the zygote will come from the mother, that is usually the egg. So the sperm will only contribute to the nuclear genome, while the mother will be also contributing to the cytoplasmic organelles, including the mitochondria, including the chloroplast, and hence including the extranuclear chromosomes. So if the extranuclear chromosomes have any kind of mutation in the mother, then the same chromosomes can be passed into progeny. In that condition, the progeny will also develop that kind of mutation. So the mutations will be passed on directly from the mother to the progeny, and there is no role of paternal extranuclear chromosomes here. So paternal chromosomes, that is the paternal cytoplasmic chromosomes, will never affect the progeny. So only if the mother's cytoplasmic chromosomes are mutated or they are affected by some kind of disorder, then the progeny will also inherit this. And this is a pure and simple definition of cytoplasmic inheritance. So cytoplasmically inherited characteristics frequently exhibit extensive phenotypic variations because no mechanism which is analogous to mitosis or meiosis ensures that cytoplasmic genes are evenly distributed in the cell division. Now, as I was telling you, one cell can contain, even most cells contain thousands of mitochondria, and each mitochondria consists of 2 to 10 copies of the similar DNA. So suppose there is only one copy of DNA out of 10 which is mutated. So this is there in the one mitochondria only. Now there are such thousands of mitochondria. Suppose we say 48 out of those 1000 mitochondria have these DNA which are mutated DNAs. So we are never knowing, and it is always, the segregation is always random. We don't know that which of these mitochondria out of these 1000 mitochondria will go into the progeny or will go into the zygote. So some of them may be mutated. In the other progenies, might be possible that the non-mutated mitochondria go. So this process is very, very random, and hence we have the higher chances that the progeny will exhibit the extensive phenotypic variations. Suppose in a cell division, some of the mitochondria have a mutated DNA while the other mitochondria have the wild type DNA. Now on a random crossing over, some of the progeny cells will receive these mutated DNA and some of the progeny cells will receive the wild type mitochondrial DNA. In that condition, all those progeny cells which are receiving the mutated DNA will also be mutated, while those progeny cells which are receiving this wild type DNA will be normal. This can also happen with the chloroplast DNA, particularly in plants. This happens. Why? Because in mammals there is only a single zygote which has to be formed, so it can be either mutated or either wild type. But in the plants there are many seeds produced, so some of them may be mutated, some of them may not be. Now there is a very peculiar example here, that is the variegation in the 4 o'clock plant. The 4 o'clock plant is also known as Mirabilis jalapa. Variegation refers to the alteration in coloring. Alteration in coloring means, suppose sometimes you are getting a green color, sometimes you are getting a white color, and sometimes you are getting a mixed color. So there is a variegation or alteration. So now here what happened is that there was a scientist whose name is Carl Correns. He worked upon the 4 o'clock plant, which is also referred to as Mirabilis jalapa. Now Correns found that when uh, the maternal branch the maternal branch was crossed over with the paternal branch, mainly the pollen grains from the paternal side, it was found that the nuclear genes of both of them were inherited into the progeny, but only the cytoplasmic genes from the maternal branches had inherited into the developing progenies. So if the maternal branch was green, that time the progeny so formed will also have the green chloroplast. Suppose we are talking about the example of chloroplast DNA. So if the chloroplast DNA is wild type, that is green colored, it is coding for the green pigment in the maternal branch, then the progeny will also have the similar kind of chloroplast DNA. If the maternal branch had this of white color is mutant form, then all of the progenies will have the similar form. And suppose if there is another case where there is the variegated maternal branch, so that time some of the progenies will receive the green colored, means that cpDNA which can code for this green pigment, and some of the progenies will get that type of cpDNA which is coding for the white pigment. So here we can get the altered progenies. So it is as simple as that, the case of cytoplasmic inheritance which was observed by Carl Correns in 1909 in the 4 o'clock plant, also referred to as Mirabilis jalapa. So that is there. Now here we can also take one of the examples. See, there is a cell division occurring here and the mitochondria are segregating randomly in the cell division. Now suppose these blue colored are the mutated mitochondria and the red ones are the normal mitochondria. There is the equal chance of getting any kind of mitochondria in the resultant cells. So results in the progeny cells that differ in their number of mitochondria with wild type and the mutated genes. Some of them may receive wild type, some of them may receive mutated, or some of them may receive both of them. So this is clear cut cytoplasmic inheritance example. Now this is what we talk about when we see the cytoplasmic inheritance. There are some of the characteristics also. First of all, this cytoplasmic inheritance can be present both in males or the females. They will always be coming from the mother. The mother cytoplasma will be giving them these traits. Secondly, they are inherited from one parent, that is usually mother. Thirdly, when we do the reciprocal cross, they often give the different results. This is very important and characteristic feature of the cytoplasmic inheritance. And lastly, they exhibit the extensive phenotypic variation even within a single family. So there is as I already told you, there would be a huge variety of phenotypes which will be observed here. So this can occur here in the cytoplasmic inheritance. So here is all of that experiment which I was discussing about, wherein all of them were crossed with the pollen, but the maternal branch in one case was white colored, maternal branch here was green colored, and here it was variegated. So for the white colored maternal branch, the progenies which were obtained were also white. For green colored we had green, and for variegated some of them were white progenies, some of them were green progenies, and some of them were mixed progenies or the variegated progenies. Now even this kind of cytoplasmic inheritance can be found in humans, but it is extremely rare case, and this is there in one of the diseases which is referred to as Leber hereditary optic neuropathy. Now patients who have this kind of disorder, that is LHON disorder, they experience rapid loss of vision in both the eyes, and it results from the death of cells in the optic nerves. And this will only be occurring when the mother is having such kind of disorder. So this will be cytoplasmically inherited to the progeny also. The loss of vision typically occurs in the early adulthood only, but it can occur anytime after the adolescence. There is much clinical variability in the severity of disease, as I talked about that the phenotypic variation is very high. And this LHON disease exhibits the cytoplasmic inheritance, that is the trait is passed from the mother to all the children, including the sons and including the daughters. In the next video we will talk about the genetic maternal effect.